Hello, everybody. It's my pleasure to be here and talk to you about the gut microbiome trajectory, immunity, and clinical implications. My name is Norbert Sprenger, and here are my disclosures. I'm an employee of the Société des Produits Nestlé in Switzerland at the Nestlé Institute of Health Sciences in Lausanne, Switzerland. Now, if we look at the microbiome, we can see something very interesting, and that is there is a very dynamic process where the microbiome changes, especially in the early life, and then it kind of stabilizes. And then maybe with older age, it seems to decrease again. So this is illustrated here in this cartoon with the wobbling. There is some variation always during adult life also, but especially we have a very dynamic process here in the early life. Now, where is this variation coming from? So this is illustrated on the right side where you see microbiome intrinsic factors. On top, you see environmental factors, host extrinsic factors, and host intrinsic factors that all can modulate and influence the microbiome and the picture that we see when we analyze the microbiome from the gut. And there is also interactions within the microbiome that are very important to be considered. But overall, we can say that age and nutrition are key drivers on how the microbiome develops and behaves over time. Now let's look at this more in the early life and the different phases in early life. So in utero, usually we are sterile. The infant does not get exposed to living bacteria, at least that's the major consensus in the scientific field, but it gets exposed to microbial metabolites that can have an effect on how the embryo develops and how the immune system in the embryo develops. Then at birth, you have a really bona fide seeding with microbes, primarily from the mother, but then also from the environment, obviously. And you can have here different phases, first 6 months, then 6 where it's exclusively breastfeeding, so that is a certain phase. Then you have mixed feeding with the introduction of complementary diet, which is very important. There is a very important step in the microbiome development. And then obviously with older age, again, the diversification that we will talk about, that is very important. Now, key benefit areas where the microbiome has a role to play is especially also in terms of pathogen resistance. So this is called colonization resistance. You can see that, for example, when you have antibiotic use, often you get more sensitive or more infected by certain pathogens that can profit now from the emptied situation in the gut. It's also important for gut barrier function, for immune competence development at large. And then what is coming up as an emerging field is also the brain and cognitive development that has some effects are reported through the microbiome, or some, at least, relationships that were shown until now. For causality, I think we need probably more research. Now, if you look at the early life determinants that, in fact, influence the microbiome development, we have, for example, maternal diet. So this can have an influence already prenatal, and there is nice data coming up here showing the influence on the developing immune system from maternal diet and their metabolites that are probably produced then by the microbes, and also by the seeding. So the mother has then certain bacteria that are to be there or not to be there, and this seeding can be influencing the later development of the microbiome. Then you have gestational age, very important. Mode of delivery with C-section as a risk factor here for microbiome development, antibiotic exposure, obviously, and the feeding mode. So now let's look at the microbiome, how it looks like over time when we look at the different developmental patterns that we see. So we can see different microbial groups that, in fact, establish in different profiles or trajectories. So you have a trajectory 1 in this report here, trajectory 2, 3, and 4, that all show a different pattern with different microbial groups as the dominant groups. For example, in trajectory 1, you see here the bifidobacterium group, which is very high early in life and then decreases while you diversify your microbiome. Now, all that gives you a picture of a dynamic, sequential, and progressive establishment and maturation of the gut microbiome. Now, this can be described also in a different, elegant way by a machine learning algorithm that comes up more and more in the literature as a model to look at the microbiome and how it's maturing. So this we call microbiome age or microbiome maturation index. And what the machine learning model does, it takes the features in the microbiome, so this can be the composition, but also the functional parts of the microbiome, functional capacity, and it tries to describe and predict the chronological age of the infant. So as we age, we get a different microbiome, and this is basically described here in this line. And then you can see here with this SHAP model how the different bacteria are important in different age groups. So for the younger ones, so below the median age of this cohort in blue, and the ones more important for the older age, above the median age, is in red. And here I want to highlight that we have many here, for example, butyrate producers, but also lactobacilli that are more important in the prediction of the older age. While for bifidobacteria, we have particularly Bifidobacterium infantis in the younger age, but we have another bifidobacterium here, Pseudocatenellatum, that is more important for the prediction of the older age. So different bacterial groups also change with time in the establishment of microbiota. Now, we don't only consider the microbiota taxonomy, but we should look at the functionality. And typically here, the different organic acids are shown in this report on the left from a Japanese group that looked in much detail over the first 2 years of life, how the different organic acids actually develop and how that reflects the different gut ecology. So you can see here, for example, the short-chain fatty acid acetate that comes up earlier and then stays more or less stable, while the propionate butyrate comes up later when you diversify the microbiome. So with diversification of the microbiome, you get more of these propionate butyrate producers. And then you have another metabolite here that is maybe very interesting. It's a succinate that is more very early organic acid in the gut ecology. Now, this is also reflected here on the right side, where you see the proportion of the different short-chain fatty acids and how they approach in the infant gut, the adult, and in this case, the mother with time. So at 1 year of age, you have more or less the profile that is also seen in mothers. Now, if we look at the microbiome in a global way, we can also define the microbiome as community types. And that is illustrated here, where you have from days 3 to 6 months of age, different community types of the microbiome that dominate the infant gut. So early on, you have particularly dominant communities with Enterobacteriaceae, and these are very quickly replaced by the community types dominated by Streptococcaceae, and then already here with Bifidobacteria, and in 2 different flavors, so dominant at around 80% Bifidobacteria or above 90% with Bifidobacteria. And you can see here the highest dominance of the Bifidobacteria, they come up around 3 to 6 months of age. So there seems to be like a bifidobacterium peak, and then as we diversify the microbiome, we get other bacterial groups in again, and this picture changes. Now let's look at some of the risk factors. And I mentioned here the C-section birth and the antibiotic use. So this is here a meta-analysis that was published on 34 different studies, and you can see here the normal trajectory of the bifidobacteria in gray, and then in the C-section born infants, the trajectory of the bifidobacteria that seems to be delayed and not reaching as high levels as in the vaginal born. And with antibiotic use overall, we can see that the bifidobacteria are quite sensitive and rather lose the presence and dominance. Now, there is some risks associated to C-section birth and antibiotic use, and I want to highlight here just the ones on allergy. There's a consistent higher risk for allergic diseases with very high sample numbers here of 1.3 million. Similar for overweight obesity, you have higher risks in large cohort studies on overweight obesity with C-section birth, and similar here for antibiotic use on allergic diseases with more frequent antibiotic use having a higher risk. And here I wanted to highlight particularly the early phase when you have antibiotic use here before 6 months of age, there seems to be a higher risk for obesity and overweight, while not so much when you have later antibiotic use. So the sensitive area is really the early phase where some priming happens. Now let's look at nutrition. So I show you here a microbiome age trajectory. And this was published based on the stratification, whether the infants get some breast milk after 3 months when these data were collected, or after breast milk, that means they had breast milk only before 3 months, but not after, or no breast milk. And you can see that there is a different trajectory. So there is a different microbiota age when they have no breast milk compared to when they have breast milk. So this is very interesting, and obviously the question is, what is in breast milk that makes the trajectory different? And I think we don't need to explain too much about the breast milk components here. So we have bioactive components and we have nutritive components, and among the bioactive components, we have the so-called human milk oligosaccharides, or HMOs, that are probably the most relevant ones here to support the microbiome development, an appropriate development. But you have also other bioactives, lactoferrin, osteopontin, glycolipids, and so on. They could all have a function also in immunity, besides the function that comes maybe through the microbiome itself. Now let's look at this in a bit more detail. So here I show you an example from a Japanese cohort. And I like this one particularly because they looked at community types again, and they see that they have a community type red, and again in this case, very dominated by Bifidobacteriaceae. And this community type here is characterized that it contains bifidobacteria that can utilize the HMOs, and particularly in this case, the fucosyl lactose as HMO. So they get this dominance because they have this type of substrate. And what happens in this community type compared to the others? Even here, another one that is very dominated by bifidobacteria, but not as high as the red one. You can see here the higher abundance of acetate compared to the other groups here. You see the lower pH as an overall measure of the different organic acids that modify the pH. And you can see also other bacterial groups like the Enterobacteriaceae that are very low in the group that has the highest bifidobacteria. So overall, there is an ecology generated when there is utilization of HMOs that looks like something very beneficial. And recently, there was also additionally other metabolites identified like indolactate, quinuronic acid, that are important for immune development, that are highly produced by bifidobacteria. And then just to come to the role of acetate here from some data from preclinical models. So this was shown to be very effective here in protecting from enteropathogenic E. coli in an animal model, in other in vitro models also from Salmonella. And then, interestingly, maybe also there is a link to the respiratory infections by respiratory syncytial virus, where there is protection with acetate. And then most recently here from this year, there's also regulation of the gut immunoglobulin A production in the gut itself through acetate. So some kind of an ecology that has a beneficial effect expected. So what did we learn when we have an intervention in formula-fed infants with HMOs? So I show you here just a snapshot of something that is published now a few years ago already. Is on the antipyretic use. We see a reduction of the risk, of the relative risk. We see a relative risk reduction also on the exposure to antibiotics, on the need of antibiotics. And then specifically what we saw is that we have a low respiratory tract infection, relative risk reduction that was significant with the HMO feeding, and specifically here in the term bronchitis. Now this trend, what we saw here was also observed in a subsequent study on cow milk protein allergic infants that got the formula with extensively hydrolyzed protein. And again, we saw a trend to the same effects. Now, what is interesting today here is the question, what are the mechanisms behind that can explain possibly these observations? And here I will show you a snapshot on the analysis that we did on the different microbial aspects to analyze at 3 months of age in this cohort or in this study. And we saw that the bifidobacterium group was particularly increased when we have the HMO feeding. So these are the two blue groups here. And you can see when we stratify C-section to vaginal-born infants, we can see that especially the C-section-born infants, they profit from the addition of the HMOs because they move here from the dark gray to this darker blue. So they increase the most the bifidobacterium population. And just to mention, in this cohort, we had about 37% C-section-born infants stratified in both, in both control and test group, the same number. When we investigated this cohort in terms of microbial community types, what we observed is that we have in the control formula fed, we have mainly a green group, which is dominated by bifidobacteria, but not as high as the most dominant microbial community type in the breastfed group, which was much higher, dominated by bifidobacteria. And you see in the test group with the 2 HMOs, we see part of the infants, they become now red, while some seem to be non-responders and they remain green. Now, when we look at just the formula-fed groups here, and we try to see whether we can now identify any link to the respiratory infection or antibiotic use through the community types, we observed only an association that was significant on the antibiotic use. And you can see that we have a protection from antibiotic use when the infant has this high dominated community type with B, the red group, is the highest bifidobacteria dominance. So this was measured here at 3 months of age, but we see the effect actually after that. So this was very interesting and the first indication that indeed we have a link between microbiota and the outcome measures that we had seen. Now, for the respiratory infections, we took a slightly different approach and we did a classification. So we asked the question, in the formula-fed infants, can we identify any microbial feature? And this included now the short-chain fatty acids and also product as the test formula, and asked the question between controls who did not experience a lower respiratory tract infection from 3 to 12 months, can we differentiate them from cases who experienced a lower respiratory tract infection from 3 to 12 months of age? And indeed, in this classification model, we can see different features that are popping up with a higher abundance here of acetate, the product. So this is what we would have expected, that the product with HMO protects from respiratory infection. And then we have fucosylated glycans, different from the 2FL that was provided in the formula. We see also then propionate butyrate, but this go more in the case. So this should not be too high early in life, and so on. And we see in the bifidobacteria group here, it also pops up on the control side. So if we look in an univariate way, you can see here the higher acetate in the controls that did not experience LRTI, while we have higher butyrate, propionate, 5-aminovalerate in the cases. And here on the bifidobacteria side, we have the sum of all the B. longums and specifically B. longum subspecies infantis that pops up on the benefit side. So there is a link maybe between those bifidobacteria and the protection from, and their metabolites and the protection from the respiratory infection that we observed. Now I just want to give you a little snapshot on the more recent study that will be presented here at abstract 60, 61, and 292. So what we found with the new trial where we have 5 HMOs tested in infant formula feeding, we saw at 3 months of age that the microbiota in general approaches the one from breastfed infants. We see particularly Bif infantis going up with the HMO feeding in the formula. And we see now, interestingly, also gut permeability markers going down, as you would have expected. And we see immune markers like the secretory IgA going up in our formula-fed infants with the HMO compared to the control. So more to come from the abstracts that you can see here. Now let's switch gears, and in the last minute or two that I have, I wanted to quickly give you a snapshot on what happens after when you introduce the weaning food and when you diversify your microbiome. So here already you can see different bacterial groups associated, for example, to atopic dermatitis, but I want to highlight again with the microbiota age, in this case expressed as a C-score, that in the healthy group you have a lower microbiota age in the early phase here, while later you seem to be higher compared to the infants who get atopic dermatitis later. So again, showing a link, and probably a link that needs to be considered in a dynamic way, that there is different history also that is important for the outcome that we measure. And this is also kind of illustrated in previous studies where a delay in the microbiota maturation at 1 year of age is related to asthma risks. Now in this study here, we come a little bit more to the nutrition part, and this will link then to the next talk that you will hear. And this is the nutritional diversification that was associated to the risk for asthma. And this is very interesting because this goes together also with diversification of the microbiota that was shown to be driven also by a food diversification. So there is a nice link between nutrition, microbiota, and later health outcomes in immune competence development. Now to summarize what I talked about, I thought I would do it in a visual way and show you here immune health-related microbiota and their functions, important at different age groups. So you can see that not all age groups have different bacterial groups. That's important. So here I just mentioned a few with Anerus stipes linked also to immune outcomes, and Fecalibacterium, important butyrate producer that is also important at later stages. We have different metabolites. I mentioned acetate butyrate that need to be probably important at different age groups, and we cannot put all into the same bucket. And then you have obviously the different nutrients that are needed. So in early life, we have breastfeeding with very dominance of HMOs, so very important here. While later, during the introduction of weaning food, you have the diversification through fibers, different fibers possibly, and other components like antioxidants, polyphenols, vitamins, and so on. And then you have different risk factors that are again very important at different age groups. And with that, I thank you very much for your attention, and I hope to see you again in real life at one point in this conference. Thank you very much.